Welcome to Forensic Serology Decoded, an overview of how serology supports investigations by identifying and characterizing biological evidence, interpreting complex serological data, and informing legal and investigative outcomes. In this presentation, we'll connect laboratory methods to practical decision-making, highlighting how careful analysis and context turn biological traces into usable evidence for investigators and courts. This slide outlines the material challenges we face when testing different substrates, from absorbent clothing and bedding to low-absorbency weapons and large vehicle surfaces. Each material affects recoverability and inhibitor presence, so labs adopt tailored extraction and sampling strategies. The graphic emphasizes that method choice must match the substrate to maximize sensitivity and preserve downstream DNA evidence. Presumptive blood tests exploit hemoglobin peroxidase activity, using reagents like TMB, Kastle-Meyer, or luminol to reveal stains quickly, either by color change or chemiluminescence. These assays are powerful for locating invisible stains, but can yield false positives from plant peroxidases or chemicals. So any presumptive result is a cue to collect samples for mandatory confirmatory testing rather than definitive proof. The brentamine, or fast red dye test, targets prostatic acid phosphatase and produces a purple precipitate in under a minute when activity is high, suggesting possible semen presence. Because cross-reactivity with vaginal acidic proteins and some bacteria can occur, this result is a presumptive indicator only and must be followed by confirmatory tests to establish sexual material beyond reasonable doubt. Semen identification relies on microscopy and alternative protein markers, with nuclear staining under high magnification providing definitive proof when sperm heads and tails are visualized. In azoospermic or vasectomized donors, microscopy may be negative, so concurrent protein markers like PSA or seminodulins are essential to achieve comprehensive coverage and avoid missed identifications. Lateral flow confirmation tests offer rapid, on-site confirmation using cassette formats that target seminodulin or PSA and hemoglobin with monoclonal antibodies. These one-step, 10-minute assays reduce cross-reactivity and provide photographic documentation, making them practical for field workflows, while still preserving material for laboratory confirmatory analysis when required. The double diffusion agar assay confirms human origin by forming a visible precipitin line when human antiserum meets human proteins, usually within 24 hours. This classical test efficiently distinguishes human from animal residues, guiding whether costly DNA testing is warranted and helping allocate investigative resources to samples most likely to yield probative human DNA. ABO antigen detection through absorption elution and mixed agglutination is useful for the roughly 80% of secretors, providing rapid exclusionary information and conserving DNA extract for subsequent testing. Beyond immediate value, these serological results can be crucial in legacy cases, where only historical serology exists, helping narrow suspects without consuming valuable genetic material. Multiplex reverse transcription PCR uses tissue-specific mRNA markers to identify body fluids and integrate that information with STR profiling for donor identification. This automated, objective workflow enables concurrent analysis of multiple fluids, standardizes results, and shortens turnaround times, streamlining casework by delivering reproducible body fluid identifications alongside DNA profiling where needed. Portable forensic triage devices miniaturize microfluidic analysis into credit card-sized platforms that perform cell lysis, filtration, and rapid colorimetric readouts while preserving a small extract for confirmatory DNA testing. These tools enable on-scene triage decisions, reduce contamination risk, and save only microliters for laboratory follow-up, improving evidence prioritization without consuming critical material. Laboratory validation and quality assurance ensure serology results are admissible and reliable through reagent lot verification, documented sensitivity and limits of detection, and measurement uncertainty reporting. Regular proficiency testing and adherence to admissibility standards, such as Daubert or Fry, provide the procedural backbone that supports judicial scrutiny and helps courts weigh serological evidence appropriately. When reporting serology results, analysts must acknowledge core limitations like mixed secretions, environmental degradation, and chemical inhibitors that affect recovery and interpretation. Reliable conclusions come from identifying and mitigating these factors, thoroughly documenting procedures and chain of custody, and avoiding overstatement of significance so findings withstand cross-examination and support just investigative outcomes.